Hello everyone, welcome to our latest chapter and our last chapter for Campbell Biology, at least for the chapters covering General Biology 1. What we're going to be looking at now is chapter 14, Mendel and the Gene Idea. And a lot of what inheritance shows is usually a simple way to represent physical traits are actually alleles, and alleles for simple cases, one is inherited from both parents, and then the combination can lead to different phenotypes, the physical traits that are actually shown, but likely the chapter will go further on later. Concept 14.1. Mendel uses the scientific approach to identify two laws of inheritance. Mendel discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. Mendel's experimental quantitative approach. Mendel's fresh approach to the study of heredity allowed him to deduce principles that have remained elusive to others. A heritable feature that varies among individuals, such as flower color, is called a character. Each variant for a character, such as purple or white color for flowers, is called a trait. Peas were available to Mendel in many different varieties. Other advantages of using peas, other than it's very easy to see difference in traits and character, short generation time, the time it takes to go from one generation of parents to offspring, then to the next offspring. Large numbers of offspring, it was easy to get multiple, and mating could be controlled very easily. Plants could be allowed to self-pollinate or could be cross-pollinated with each other. Such as the technique diagram shown here, where the parental generation is represented P, and simply the stamens and carpel, either from two different plants, could be used to fertilize. And then the results, essentially taking a purple flower plant and then the plant that had the white flower trait, all the first filial generation of offspring, that'd be F1, they all showed up to be purple. That's indicating that purple was a dominant trait. Mendel chose to track only those characters that occurred in two distinct alternatives. He also started with varieties that were true breeding, plants that produce offspring of the same variety when they self-pollinate, essentially keeping things very simple. So as not to have so many different genetic factors, confounding factors to keep track of. In a typical experiment, Mendel mated two contrasting true breeding varieties, a process called hybridization. The true breeding parents are called the P generation. The hybrid offspring of the P generation are called the F1 generation. When F1 individuals self-pollinate or cross-pollinate with other F1 hybrids, the F2 generation is produced. The law of segregation. In the 1800s, the explanation of heredity was the blending hypothesis. When Mendel crossed contrasting true breeding white and purple flowered pea plants, all of the F1 hybrids were purple. The result was not predicted by the blending hypothesis. In other words, taking an all white plant and an all purple plant, there should be flowers that are lightly purple. That was the blending hypothesis, which was not the case. What Mendel's first generation of flowers achieved was a purple plant and a white plant hybridized, led to an entire generation of equally purple plants as one of their parents. When Mendel crossed the F1 hybrids, many of the F2 plants still had purple flowers, but some had white flowers instead. Mendel discovered a ratio of about 3 purple flowers to 1 white flower in the F2 generation. So according to his experiment, taking the parent generation, cross-pollinating a purple flower and a white flower together, the seeds from that combination led to the F1 generation of hybrids where all of them were purple. And then pollinating just among the F1 generation, that led to 705 purple flowered plants and 224 white flowered plants, which is very incredible. This probably took Mendel quite a good many years, or months, or whatever. And then this graphic, if you have your e-text, you would be able to see this graphic, which is a good point to stop and just ask if you guys would like to like the video, subscribe to the channel, things like that, just to see where this channel grows. That's awesome. And of course, leave a comment wherever you are, whatever college class you're taking or in high school in preparation, and leave a comment. I'd be interested to see what you guys are up to, guys and gals. Here, the principle is using a Punnett square, which is a way of representing the possible allele that could be inherited from one parent and the possible allele that could be inherited from another parent. Mendel reasoned that only the purple flower factor was affecting flower color in the F1 hybrids. Mendel called the purple flower color a dominant trait and the white flower color a recessive trait. The factor for white flowers was not diluted or destroyed because it reappeared in the F2 generation, disproving the blending hypothesis for being the end-all be-all. Mendel observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 traits, what Mendel called a hereditable factor is what we now call a gene. The results of Mendel's crossed F1 crosses for 7 characters in pea plants. What he looked at was flower color between purple and white, seed color between yellow and green, seed shape between round and wrinkled, pod color between green and yellow, pod shape between inflated and constricted, flower position, axial along the stem versus terminal at the end, and stem length, essentially tall versus dwarf plants. And the ratios shown on the right for those various factors. Sometimes about 3 to 1, so 1 out of every 4. For many of those traits, actually appears that it was 3 to 1 for each one, which gave the impression of a 25% chance to see the recessive trait and a 75% chance to see the dominant trait. Mendel's model. Mendel developed a model to explain the 3 to 1 inheritance pattern he observed in the F2 offspring. 4 related concepts make up this model. These concepts can be related to what we know about genes and chromosomes. First, alternative versions of genes account for variations in inherited characters. For example, the gene for flower color in pea plants exists in 2 versions, one for purple flowers and one for white flowers. These alternative versions of a gene are called alleles. Each gene resides at a specific locus on a specific chromosome. So what we see here is representation. On top part, the allele for the purple flowers, and the locus and then on the bottom, the allele for white flowers. And they're existing on the same locus for the flower color gene. And they can be a pair of homologous chromosomes. Essentially, they're the same chromosome, and production of enzyme that helps synthesize the purple pigment, or the absence of that enzyme, therefore no purple pigment. One allele results in sufficient pigment for purple flowers. Second, for each character, an organism inherits two alleles, one from each parent. Mendel made this deduction without knowing about chromosomes. Very impressive. The two alleles at a particular locus may be identical, as in the true breeding plants of Mendel's P generation. Or the two alleles at a locus may differ, as in the F1 hybrids. Third, if the two alleles at a locus differ, then one, the dominant allele, determines the organism's appearance. The other, the recessive allele, has no noticeable effect on appearance. In the purple color, or in the flower color example, the F1 plants had purple flowers because the allele for that trait is the dominant version of that character. Fourth, the law of segregation. The two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes, different egg cells, different sperm cells. Thus, an egg or a sperm gets only one of the two alleles that are present in the organism. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. The model accounts for the 3-to-1 ratio observed in the F2 generation of Mendel's crosses. Possible combinations of sperm and egg can be shown using a Punnett square. A capital letter represents a dominant allele, and a lowercase letter represents a recessive allele. Shown here, going from the parent generation to the F1 filial 1 generation to the filial 2 generation. What we see here are essentially a Punnett square, where one parent, one F1 parent being heterozygous, has a dominant trait and a recessive trait, and then the other parent has a dominant trait and a recessive trait in this case. And then the Punnett square is just crossing together each one, where in this first square, that offspring, that F2 offspring, inherited two dominant purple alleles, therefore overall it would show purple. And then in one case, one parent plant gives it a dominant P trait, and then the other gives it a lowercase or recessive trait. Therefore, the dominant purple gene still shows up, just like in the other case. But then there's only one situation in this particular example where there could be one offspring that inherits just one recessive allele from both. Therefore, it would not express that dominant trait of purple. And that is how a Punnett square works. You take both parents, and you represent their alleles that they could give off to the offspring, and then you just cross to see what traits you end up getting. Homozygous dominant, heterozygous, homozygous recessive. And speaking of all those terms, let's talk about useful genetic vocabulary. An organism with two identical alleles for a gene is called a homozygote. It is said to be homozygous for the gene controlling that character. An organism with two different alleles for a gene is a heterozygote, and is said to be heterozygous for the gene controlling that character. Unlike homozygotes, heterozygotes are not true breeding. That means that they're not going to just only have offspring 100% of the time that it has the same expressed trait as what's being seen in the heterozygote. If there's a dominant trait, perhaps two heterozygotes will give a offspring that inherits just the recessive trait. An organism's traits do not always reveal its genetic composition. Therefore, we distinguish between an organism's phenotype, or the physical appearance, and its genotype, the genetic makeup. In the example of flower color in pea plants, capital PP and capital P lowercase p plants have the same phenotype or purple, but they have different genotypes. For example, if we look at 3 plants that were found to be purple, one of them might have been homozygous dominant, only inheriting the dominant allele from each parent. 2 of them end up being heterozygous, inheriting a dominant allele and a recessive allele, but expressing the dominant allele. And then 1 offspring with white flowers was homozygous but recessive. It only inherited the recessive alleles. An individual with the test cross, an individual with the dominant phenotype, could be either homozygous dominant or heterozygous. It's actually hard to tell just from the phenotype what the genotype is. To determine the genotype, we carry out a test cross, breeding the mystery individual with a homozygous recessive individual. If any offspring display the recessive phenotype, the mystery parent had to be heterozygous because it had the opportunity to give a recessive allele to that plant, leaving it recessive. For example, shown here, the technique used is the plant homozygous dominant or heterozygous, fertilizing that plant or breeding it with a homozygous recessive plant leads to the following example. On the left, let's say that plant was homozygous dominant. Well, basically, if you keep having offspring that only express the dominant trait, then that is a good indicator that it is homozygous dominant. But if in any case the recessive character gets displayed, then you know that that's only possible by inheriting both a recessive allele from both parents. One parent we know can only give the recessive allele, so to show up as recessive, the unknown parent had to be heterozygous. The law of independent assortment. Mendel derived the law of segregation by following a single character. The F1 offspring produced in the cross were monohybrids, meaning they were heterozygous for one character. A cross between such heterozygotes is called a monohybrid cross. Mendel identified his second law of inheritance by following 2 characters at the same time. Crossing 2 true breeding parents differing in 2 characters produced dihybrids in the F1 generation, heterozygous for both characters. A dihybrid cross, a cross between F1 dihybrids, can determine whether 2 characters are transmitted to offspring together as a unit or independently. For example, is seed shape and seed pod color, do they keep showing up together over and over again? Or can a plant be recessive for one and dominant for the other? Or so on. So what we usually represent that is with more capital and lowercase letters. In this case, looking at a plant that has the yellow seed color, the dominant color, and the round seed shape, versus the green color represented by lowercase y and the wrinkled shape represented by lowercase r. The F1 generation, in this case, would inherit a dominant y and lowercase y, but the other trait for seed shape, not seed color, well, a dominant trait and a recessive trait, the dihybrid. And then crossing F1 generations, hypothesis of dependent assortment, meaning that dominant traits always follow dominant traits. Yellow color, round color, yellow color, round color, green color, wrinkled, green color, wrinkled. This would indicate if dependent assortment was true, that these separate genes were not independently heritable. It would be a 3-to-1 example. The expected phenotypic ratio. However, the hypothesis of independent assortment would follow this. There may be a quarter of the time where that parent gives both dominant alleles, and there may be 2 quarters where, 1 quarter where the dominant seed color and the recessive seed shape is given, the recessive seed color and the dominant seed shape, or both recessive characters, and on the other side as well. And what this ends up giving is the dominant yellow color and the dominant round color in a 9-to-16 ratio, a recessive green color but a dominant round shape in the 3-to-16 ratio, wrinkled recessive seed shape but dominant yellow color 3/16, whereas wrinkled recessive wrinkled shape and recessive green color 1/16. So phenotypic ratio of 9-to-3 to 3-to-1. And the actual results that were found by Mendel was, out of all the plants, 315 had dominant round and yellow seeds. 108 had the dominant round shape but the recessive green shape. 101 had the dominant yellow color but the recessive wrinkled shape. And 32 had the recessive wrinkled shape and green color, which ended up being about a phenotypic ratio of 9-to-3 to 3-to-1, which goes to support that these individual traits, the yellow or green color and the round or wrinkled shape, they are independent. Basically, you can inherit the dominant version of one, and maybe the dominant or recessive version of the other. You're not just going to see dominant traits always together, and you're not just going to see recessive traits always all together. Using a dihybrid cross, Mendel developed the law of independent assortment. It states that each pair of alleles segregates independently of any other pair of alleles during gamete formation. This law applies only to genes on different non-homologous chromosomes, or those far apart on the same chromosome. Genes located near each other on the same chromosome tend to be inherited together. Concept 14.2. Probability laws govern Mendelian inheritance. Mendel's laws of segregation and independent assortment reflect the rules of probability that apply to tossing coins or rolling dice. When tossing a coin, the outcome of one toss has no impact on the outcome of the next toss. In the same way, the alleles of one gene segregate into gametes independently of other gene alleles. The multiplication and addition rules applied to monohybrid crosses. The multiplication rule states that the probability that 2 or more independent events will occur together is the product of their individual probabilities. Probability in an F1 monohybrid can be determined using the multiplication rule. Segregation in a heterozygous plant is like flipping a coin. Each gamete has half a chance of carrying the dominant allele and half a chance of carrying the recessive allele. The addition rule states that the probability that any one of 2 or more mutually exclusive events will occur is calculated by adding together their individual probabilities. The rule of addition can be used to figure out the probability of an F2 plant that has a monohybrid cross. An F2 plant from a monohybrid cross will be heterozygous rather than homozygous, such as crossing two heterozygous individuals for one specific trait, segregation of alleles into the eggs and segregation of alleles into the sperm. Essentially flipping the coin, there's half a chance of from one parent inheriting the dominant trait or the recessive trait, and then for the other parent, there's half a chance of inheriting the recessive trait, half a chance of inheriting the recessive trait. And then what happens is we multiply those probabilities. A half times a half is a quarter. Probability of getting just the dominant genotype, then there's a quarter chance of these two parents having a dominant heterozygous. There is a quarter chance of these two parents, 1/2 times 1/2, to yield a heterozygous offspring, and then 1/2 chance multiplied by 1/2 chance yields a quarter chance of having a homozygous recessive. And then for the same genotype, we just add those probabilities together. So a quarter to a half to a quarter. Solving complex genetics problems with the rules of probability. We can apply the rules of probability to predict the outcome of crosses involving multiple characters. A multi-character cross is equivalent to 2 or more independent monohybrid crosses occurring simultaneously. In calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. So the probability of inheriting the probability for the dominant yellow color for a homozygous dominant, and then the probability of the seed shape for a homozygous dominant. There's a quarter probability of giving just dominant traits, and then there's a quarter chance for the yellow color and a quarter chance for the dominant round shape. Quarter times a quarter equals 1/16. The probability for YY, RR, essentially, or capital Y, lowercase y, capital R, that would be 1/2 times a quarter equals 1/8 chance of those two parents yielding a homozygous dominant round seed that is also heterozygous for yellow color. And then the other probabilities, essentially, as you add more and more traits, these independently inheritable traits, you just multiply their Punnett square probabilities together to get the overall probability of an offspring having all of that genotype. And then when we have different probabilities, and we're looking at, say, the chance of inheriting at least 2 recessive traits, such as lowercase p, lowercase y, lowercase p, lowercase y, lowercase r, at least 2 lowercase letters. Adding up all those chances together, you get 6/16, which just simplifies to 3/8. So the individual opportunities, you can add those probabilities together. Concept 14.3. Inheritance patterns are often more complex than predicted by simple Mendelian genetics. The relationship between genotype and phenotype is rarely as simple as in the pea plant characters Mendel studied. Many heritable characters are not determined by only one gene with 2 alleles. However, the basic principles of segregation and independent assortment apply even to more complex patterns of inheritance. Extending Mendelian genetics for a single gene Inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations: when alleles are not completely dominant or recessive, when a gene has more than two alleles, when a gene produces multiple possible phenotypes. Degrees of dominance. Complete dominance occurs when phenotypes of the heterozygote and dominant homozygote are identical. That means that whether it's a dominant homozygous or heterozygous, if the dominant trait is being expressed, it looks the same no matter what the genotype is between those two. Incomplete dominance, the phenotype of F1 hybrids is somewhere in between the phenotypes of the two parental varieties. That would have been if a dark purple plant and a white plant lead to offspring that are light purple. This is seen in some other plants. Codominance, the two dominant alleles affect the phenotype in separate, distinguishable ways. Such as this case, a different plant, we had the P generation for red color. C capital R, C capital R. And then the white color, C capital W, C capital W. When they're crossed together, this ended up leading to a pink plant, so a slight in between red and white. And the gametes after that of this F1 generation can either just be inheriting the red trait or just be inheriting the white allele, I should say. And then the F2 generation made from crossing these pink flowers. And there was a core chance of the sperm and the egg cell just giving off the red allele, a core chance of just having a pink flower, another core chance of having a pink flower, and a quarter chance of just having the recessive white color. The relationship between dominance and phenotype. In the case of P-shape, the dominant allele codes for an enzyme that converts an unbranched form of starch in the seed to a branched form. The recessive allele codes for a defective form of the enzyme, which leads to an accumulation of unbranched starch. This causes water to enter the seed, which then wrinkles as it dries. Tay-Sachs disease is a fatal inherited disorder. A dysfunctional enzyme causes an accumulation of lipids in the brain. At the organismal level, the allele is recessive. At the biochemical level, the phenotype, that is, the enzyme activity level, is incompletely dominant. At the molecular level, the alleles are codominant. Frequency of dominant alleles. Dominant alleles are not necessarily more common in populations than recessive alleles. One baby out of 400 in the United States is born with extra fingers or toes. The condition is called polydactyly, and it is caused by a dominant allele. Found much less frequently in the population than the recessive allele. Multiple alleles. In most genes exist in populations in more than two allelic forms. For example, the four phenotypes of the ABO blood group in humans are determined by three alleles for the enzyme that attaches A or B carbohydrates to red blood cells. Capital A, dominant B, or lowercase I, which would be for blood type O, lacking the A and B carbohydrates. The enzyme encoded by the dominant A allele adds the A carbohydrate whereas the enzyme encoded by the B allele adds the B carbohydrate. The enzyme encoded by the I allele adds, by the lowercase I allele, adds neither carbohydrate. So, in chart A, the three alleles for the ABO blood groups and their carbohydrates. Dominant A attaches A carbohydrates to the red blood cell. Dominant B attaches B carbohydrates, and lowercase I attaches none. Therefore, in chart B, blood group genotypes and phenotypes, two dominant A's, or a heterozygous dominant A. The red blood cell with surface carbohydrates has the A carbohydrate, so blood type, the phenotype that would be observed is A. A combination of just dominant B's, homozygous or heterozygous, the red blood cells would still have the B carbohydrate. A mix of both, dominant A and dominant B, yields the red blood cells having both carbohydrates on them, which leads to the AB blood type. And then being homozygous recessive, the blood cell does not have either A or B carbohydrates. Therefore, that would be blood type O. Most genes have multiple phenotypic effects, a property called pleiotropy. For example, pleiotropic alleles are responsible for the multiple symptoms of a certain hereditary diseases, such as cystic fibrosis and sickle cell disease. Extending Mendelian genetics for two or more genes. Some traits may be determined by two or more genes. In epistasis, one gene affects the phenotype of another due to interaction of the gene products. In polygenic inheritance, multiple genes independently affect a single trait. Epistasis. In epistasis, expression of a gene at one locus alters the phenotypic expression of a gene at a second locus. For example, in Labrador retrievers, in the dog breed, and many other mammals, coat color depends on two genes. One gene determines the pigment color, with alleles for either dominant black or recessive brown. The other gene, with alleles for dominant color or recessive no color, determines whether the pigments will be deposited in the hair. In heterozygous black labs, genotype being heterozygous for both B and E, if they are mated, we might expect a dihybrid F2 ratio of 9:3 to 3:1. However, a Punnett square shows that the phenotypic ratio will be 9 black to 3 chocolate to 4 yellow labs. Epistatic inheritance, or epistatic interactions, produce a variety of ratios, all of which are modified versions of 9:3 to 3:1. For example, looking at a cross between two heterozygous black labs, what we end up seeing is a variety combination of mostly black labs, some chocolate labs, and then some golden retrievers. Or golden color. Polygenic inheritance. Quantitative characters are those that vary in the population along a continuum. Quantitative variation usually indicates polygenic inheritance, an additive effect of two or more genes on a single phenotype. Height is a good example of polygenic inheritance. Over 180 genes affect height. Skin pigmentation in humans is also controlled by many separatable inherited genes. In this case, this would be For skin color, getting a wide range of possible combinations for various hues of skin or shades. I'm not sure what the proper artistic term would be. Which is why you may end up seeing kids, grandkids, great-grandkids that are unusually a different shade, where all the parents, grandparents, great-grandparents seem to be the same color, because all these different genes may get spread along each generation until eventually you get a combination that leads to a sharply different color from what most in that family ends up being. Nature and nurture, the environmental impact on phenotype. Another departure from simple Mendelian genetics arises when the phenotype for a character depends on environments as well as genotype. The phenotypic range is broadest for polygenic characters, traits that depend on multiple genes combined with environmental influences are called multifactorial, such as hydrangeas grown in basic soil will be more pink, where hydrangeas of the same genetic variety grown in acidic soil with free aluminum can come out more purple or blue. A Mendelian view of heredity and variation. An organism's phenotype includes all aspects of its physical appearance, internal anatomy, physiology, and behavior. An organism's phenotype reflects its overall genotype and unique environmental history. Concept 14.4. Many human traits follow Mendelian patterns of inheritance. Humans are not good subjects for genetic research. The generation time is too long. Parents produce relatively few offspring. Breeding experiments are unacceptable, very immoral and unethical. However, basic Mendelian genetics endures as the foundation of human genetics. There is pedigree analysis. In human genetics, geneticists analyze the results of human mating that have already occurred. A pedigree is a family tree that describes the inheritance of a trait across generations. Looking at this, we have squares representing a male individual, circles representing a female individual. If the square or circle is shaded in, then they have that trait. And a horizontal line represents mating between individuals, and offshoots represent offspring in birth order. The first born on the left, and then the last born on the right. For chart A, looking at is widow's peak a dominant or recessive trait? What we have here is a combination of a heterozygous individual and a homozygous recessive individual, and it appears that 2 of the kids end up showing that trait, whereas another situation, 1 kid ended up having that widow's peak expressed and 1 didn't. And then the cross between 2 heterozygotes still led to a probability of seeing that widow's peak, whereas a very little probability of seeing the 0 widow's peak, indicating that widow's peak was actually on a dominant trait. Now in C is the inability to taste a chemical called PTC, a dominant or recessive trait. Well, heterozygotes that lack the trait ended up having some offspring that still lacked that trait and were heterozygote, and then there were a cross between a heterozygote and a homozygous recessive that had that trait and ended up yielding another heterozygote that did not express the trait. But then the cross between two heterozygotes, there was one that ended up inheriting just the recessive alleles and was able to express the lack of taste of PTC. Therefore, let's see. Therefore, the ability to taste it was always the dominant trait, whereas the lack of ability to taste it was always the recessive quality. And then if you have your e-text, you can use this animation called simplified cross of one character in humans. In this case, freckles and no freckles. Once again, if you would like, by all means, like the video, subscribe to the channel, and leave a comment if you feel so inclined, none of which are necessary, but they're just fun to see. Moving on. Pedigrees can be used to make predictions about future offspring. We can use this multiplication and addition rules to predict the probability of specific phenotypes. Recessively inherited disorders. Many genetic disorders are inherited in a recessive manner. These range from relatively mild to life-threatening. The behavior of recessive alleles. Recessively inherited disorders show up only in individuals who are homozygous for the allele. Carriers are heterozygous individuals who carry the recessive allele but are phenotypically normal or healthy. Most individuals with recessive disorders are born from carrier parents. Albinism is a recessive condition characterized by a lack of pigmentation in skin and hair, such as two parents that are phenotypically normal, but they're heterozygous. There is 3 chances out of 1 that at least one dominant allele will be inherited to express that trait, and then 1 out of 4 for just the recessive trait to show up due to inheriting both recessive alleles for albinism. If a recessive allele that causes a disease is rare, it is unlikely that 2 carriers will meet and mate. Consanguineous mating, that is, meeting between close relatives, can increase the chance that both parents of a child carry the same rare allele if that rare allele shows up quite a bit in that mini-group family. Most societies and cultures have laws or taboos against marriages between close relatives, typically between siblings, parents, grandparents, and sometimes as far as aunts, uncles to nieces and nephews. Cystic fibrosis is the most common lethal genetic disease in the United States, striking 1 out of every 2,500 people of European descent. Cystic fibrosis allele results in defective or absent chloride transport channels in plasma membranes, leading to a buildup of chloride ions outside the cell. Symptoms include mucus buildup in some internal organs and abnormal absorption of nutrients in the small intestine. If left untreated, cystic fibrosis can cause death by the age of 5. Daily doses of antibiotics to stop infection and physical therapies can prolong life. In the United States, more than half of those with cystic fibrosis now live well into their 40s. Sickle cell disease, a genetic disorder with evolutionary implications. Sickle cell disease affects 1 out of 400 African Americans. It is caused by the substitution of a single amino acid in the hemoglobin protein in red blood cells. In homozygous individuals, all hemoglobin are abnormal or sickle cell shaped. Symptoms include physical weakness, pain, organ damage, and even paralysis. Heterozygotes said to have the sickle cell trait are usually healthy but may suffer some symptoms. About 1 out of 10 African Americans has sickle cell trait, an unusually high frequency. Heterozygotes are less susceptible to the malaria parasite, though, so there is an advantage to being heterozygous in regions where malaria is common. Here we see the locus for the sickle cell allele, and then the sickle cell hemoglobin proteins that are formed from that genetic code. Low oxygen levels are carried, part of a fiber of sickle cell hemoglobin proteins sticking together, leading to the sickle cell shape for the red blood cell. Long fibers cause red blood cells to be sickle shaped, and then you get your sickle cell disease. That would be for chart A, homozygote with sickle cell disease, weakness, anemia, pain and fever, and organ damage. But in the case of chart B, heterozygote with sickle cell trait, some symptoms when blood oxygen is very low, but also reduction of malaria symptoms. You have both the allele for sickle cell and the allele for typical cell shape. You'll get both types of hemoglobin, sickle cell and normal hemoglobin proteins, and you'll have a mix of each, and therefore you'll have a mix of typical red blood cells and sickle cell red blood cells. But you'll still see the sickle cell trait expressed, if not the severity of the disease for homozygotes. Dominantly inherited disorders. Some human disorders are caused by dominant alleles. Dominant alleles that cause a lethal disease are rare and arise by mutation. Achondroplasia is a form of dwarfism caused by a rare dominant allele. For example, let's take the parents, one that has homozygous dwarfism, and then the other that has the normal phenotype for non-dwarfism. In this case, the only offspring that would be dwarfs are those that inherit the dominant allele from the one parent, whereas there's just a half a chance of inheriting just two recessive typical height. The timing of onset of a disease significantly affects its inheritance. Huntington's disease is a degenerative disease of the nervous system. The disease has no obvious phenotypic effects until the individual is about 35 to 40 years old. 35 to 40. Once the deterioration of the nervous system begins, the condition is irreversible and unfortunately fatal. There is a test that can detect the presence of the Huntington's allele in an individual's genome. Some individuals with a family history of Huntington's disease choose to be tested for the allele. Others decide that it would be too stressful to find out and make the personal choice to not know. Multifactorial disorders. Many diseases such as heart disease, cancer, alcoholism, and mental illnesses have both genetic and environmental components. No matter what our genotype, our lifestyle has a tremendous effect on phenotype. Genetic testing and counseling. Genetic counselors can provide information to prospective parents concerned about a family history for a specific disease. Fetal and newborn testing can also reveal genetic disorders. Counseling based on Mendelian genetics and probability rules. Suppose a couple both have a brother who died from the same recessively inherited disease. A genetic counselor can help determine the risk that this couple will have a child with that disease. It is important to remember that each child represents an independent event in the sense that its genotype is unaffected by the genotypes of older siblings. If both members of the couple had a sibling with the recessively inherited illness, both of their parents were carriers. Thus, each has a 2/3 chance of being a carrier themselves. If both are carriers, there is a 1/4 chance of each child having the recessive illness. The overall probability of them having a child with this illness would be 2/3 multiplied by 2/3 multiplied by 1/4, or 1/9 chance. Tests for identifying carriers. For a growing number of diseases, tests are available that identify carriers and help define the odds of having an affected child more accurately. The tests enable people to make more informed decisions about having children. However, they raise other issues, such as whether affected individuals fully understand their genetic test results and how the test results are used. Fetal testing. In amniocentesis, the liquid that bathes the fetus is removed and tested for certain genetic disorders. In chorionic villus sampling, CVS, a sample of the placenta is removed and tested. Other techniques, such as ultrasound, allow fetal health to be assessed visually in utero. Here's an example of amniocentesis, where the amniotic fluid is withdrawn during an ultrasound, and then through centrifugation, the amniotic fluid is separated from the fetal cells, and the fetal cells can be cultured, and then the karyotype can be determined. And on the fluid, genetic, including DNA-based and biochemical tests can easily be performed. And chorionic villus sampling, essentially it's just taking fluid, fetal cells, to perform genetic DNA testing and to perform cell cultures to get karyotypes. And then we have the ultrasound of a fetus. Newborn screening. Some genetic disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the United States. One common test is for phenylketonuria, PKU, a recessively inherited disorder that occurs in 1 out of every 10,000 to 15,000 births in the United States. The number of conditions that can be tested in newborns is over 100 and ever increasing. And now we have our variety. An example of just different individuals with a variety of traits, from curly hair, coiled hair, wavy hair, straight hair, dark, light, and various other qualities as well. Here we just have our phenotype and then the number of dark skin alleles by R.A. Strom, A Golden Age of Human Pigmentation Genetics, from Trends in Genetics, issue 22, pages 464 to 468, published in the year 2006. And we just have some more examples of segregation of alleles into sperm or egg. Examples of the relationship of alleles of a single gene, such as complete dominance of one allele, incomplete dominance of either allele, codominance, multiple alleles, and Pleiotropy. Chart showing epistasis and polygenic inheritance. That graph used for widow's peak, showing that it was a dominant trait, and looking at the expression of homozygotes with sickle cell alleles. And then just a cat. And then just a example system of different inheritance factors. Of what I assume is a figure of a family at a beach before color was discovered. And yes, that concludes our video. This concludes our chapter readings for our audio narrated chapter readings for General Biology 1, college level General Biology 1. If there are any other chapters you would like discussed or reviewed on this channel, please suggest them by all means, or another textbook as well. And if you made it to this far in the video, as always, consider liking the video, subscribing to the channel, and simply just leaving a comment about the value you got from this, and where you would like to see other content. In any case, wishing you all the best in your class, or your classes, wherever they may be, and of course, peace.